Hey everyone, welcome to our first content lecture in medical immunology. We're starting broad because everything we do for the rest of the semester is really going to build on the framework that we establish here. When we eventually talk about autoimmune disease, hypersensitivity, immunodeficiency, transplantation, serological testing, or infectious disease, we're really asking variations of the same questions. And that's, what did the immune system recognize? How did it respond? Which cells were involved? And was the response appropriate? For part one, I want you thinking less about memorizing isolated facts and more about building a map of the immune system. So these objectives should give you a good idea of what I expect you to be able to do with this material. You'll need some historical context, but our major focus as a whole is going to be comparing innate and adaptive immunity, recognizing the major immune cells and what they do, understanding where lymphocytes develop and become activated, and beginning to apply those concepts clinically. So, when you're studying, I don't want you to just ask yourself, can I define what a neutrophil is? I want you to ask yourself, if neutrophils are elevated in the patient, what does that tell me about what's happening? That's the level that we're really building toward. Now, for these key terms, don't try to memorize this slide right now by any means. Think of this as your vocabulary map. Some of these terms will make complete sense today, whereas others will become more meaningful as we move through this unit as a whole. You'll notice that some terms describe systems like innate and adaptive immunity, and some describe cells like neutrophils and lymphocytes. Others describe how those cells recognize or communicate about threats like PAMPs, PRRs, cytokines, MHC, and complement. By the end of this unit, I really want you to be able to connect these terms rather than just define each one in isolation. So before we talk about what we know today, it's helpful to really see how we got here. So humans were manipulating immune responses long before we understood cells, antibodies, antigens, microorganisms. And immunology really developed from observation first and mechanism second. So, the early history of immunology is essentially people recognizing that exposure can really change future susceptibility to disease. As early as the 15th century, people were deliberately exposing individuals to material from smallpox lesions in an attempt to prevent severe disease. Jenner later used cowpox material to protect against smallpox, and Pasteur expanded this idea by developing attenuated vaccines. They didn't have our current understanding of lymphocytes or immunologic memory, but they were demonstrating one of the most important characteristics of adaptive immunity, which is previous exposure changes the response to future exposure. Then researchers really started asking why. Why does this happen? Metchnikoff observed phagocytosis, which giving us evidence that cells physically participate in host defense. Von Behring demonstrated that something in serum could neutralize bacterial toxins, and now we have evidence for a soluble or humoral component. And then Wright helps connect these ideas by showing that substances in serum can coat organisms and make them easier for phagocytic cells to ingest. And that's an important theme for this entire course. The immune system isn't just a collection of independent parts, it's components working beautifully together. So now we're going to move from the history into the system itself. As we go through this section, I really want you to keep one basic question in mind. If a microorganism enters the body right now, what happens next? And we're going to build that answer piece by piece. At its simplest, immunology is the study of how a host reacts to a foreign substance. One important term right here is antigen. An antigen is something capable of being recognized by the immune system and provoking an immune response. That doesn't automatically mean a dangerous microorganism. Pollen can act as an antigen. Components of bacteria and viruses can act as an antigen. So we really need to separate the ideas of foreign, recognized, and harmful because those aren't always the same thing. Here's where immunology gets interesting. An immune response isn't automatically a good response. Too little response can leave someone susceptible to infection, whereas too much response can damage tissues. A response directed at something harmless can produce an allergy, and then a response against our own tissues can produce autoimmunity. An appropriate immune response against tissue that doesn't belong to us becomes a problem when that tissue happens to be a transplanted organ. So, throughout this entire semester, we're really going to be studying balance. This table really shows you why immunology extends far beyond infection. Yes, the immune system protects us from microorganisms, but it also participates in tumor surveillance, tissue repair, inflammation, transplantation, and responses to newly introduced proteins. And notice the duality here. Immune activity can protect us from infection, but immune dysfunction can cause disease. Immune cells can help repair damaged tissues, but inflammation can also damage tissue. That idea, the protective versus pathologic immunity, is really going to come up repeatedly throughout this semester. Again, it's all about that balance, right? So now we're going to organize everything into two major arms. We're going to have the innate immunity and the adaptive immunity. I don't want you to think of these two immune systems as operating separately, right? Again, think of them as two interconnected strategies. One responds quickly and recognizes broad patterns, where the other develops a highly specific response and can generate that memory. And they communicate constantly. So, innate immunity is what you have available right now. It doesn't need to encounter a specific organism previously. It recognizes general features associated with danger or microorganisms, and it responds very rapidly. So that's why we describe it as non-specific. If bacteria enters through a wound, your innate system doesn't just sit around asking, have I met this exact strain before? It recognizes that something doesn't belong there, and then it responds. And traditionally, we say innate immunity does not generate immunological memory. One way innate cells recognize microbes is through the pattern recognition receptors, or PRRs, which recognize common microbial patterns called PAMPs. So I want you to think the PAMP, PAMP, is the pattern on the pathogen, whereas the PRR is the receptor on the host cell that recognizes that pattern. And we're going to talk a little bit more about the PAMPs and PRRs in part 2 of the lecture. So, adaptive immunity is different because it's antigen specific. Our B and T lymphocytes carry special receptors capable of recognizing particular antigens. So, when the appropriate lymphocyte encounters its antigen, it receives the appropriate signals. That population can expand and participate in this immune response. It says, hey, come to the party. The other major difference is memory. So, after exposure, some lymphocytes persist as memory cells. And if the antigen appears again, the response is going to be faster and stronger. And that principle eventually brings us back to vaccination. Again, if I cut my hand and bacteria enters the wound, which is going to respond first, the innate or the adaptive, right? We'll also talk about the different, a little bit more into the semester, we'll talk about the different immunoglobulins. And when we talk about the first response, that is our immunoglobulin M, or IgM, that's produced in abundance first. So first exposure is IgM. And then that secondary or memory response is immunoglobulin G, or IgG. And that one is going to be produced in abundance upon any secondary or subsequent responses. And I always like to tell students that the way I like to remember that IgG is like the memory reactors is because it's G, because it holds a grudge. It's going to remember that pathogen or that antigen, that foreign body, next time it is introduced into the immune system. All right. So now that we know the two major strategies, we really need to meet the cells that are carrying them out. And this is where hematology is married to immunology. So hopefully you have learned or are currently learning how to identify these cells morphologically. But then we're also going to attach the immune function to that morphology. So if you've taken hematology, you're ahead of the curve or being able to identify these cells and their function. If you're currently taking immunology or hematology with immunology, then hopefully this helps make it stick a little bit more. So all of these immune cells ultimately trace back to the hematopoietic stem cells within the bone marrow. Those stem cells differentiate broadly into either myeloid or lymphoid lineages. And for our purposes, I just want you to remember the big picture, okay? Myeloid precursors give us granulocytes, monocytes, and dendritic cells, whereas the lymphoid precursors are giving rise to the lymphocytes. You do not need to memorize every arrow on this diagram today. I just really want you to understand the family tree. So here's where our innate versus adaptive distinction gets a little messier. So our innate immunity is going to also include the physical epithelial barriers, tissue macrophages, dendritic cells, mast cells, granulocytes, monocytes, natural killer cells, and other innate lymphoid cells, whereas adaptive immunity is primarily mediated by the B and T lymphocytes. But again, these groups interact, right? They're not independent, they interact together. So tissue resident innate cells can recognize microbes and help initiate the responses that eventually activate the adaptive immunity. So we're building a network again, right? Not two separate lists. Cells also need places to develop and to interact. So I want you to think how primary lymphoid organs are where our lymphocytes mature. So I always like to remember that B cells are B because they mature in the bone marrow, whereas T cells mature in the thymus. Our secondary lymphoid organs are where the mature lymphocytes will then encounter antigens and become activated. The distinction is very testable, but I really want you to understand the logic rather than memorizing the words. So, again, primary preparation, secondary activation. So this slide is going to be our roadmap for the next several slides. We're going to divide the cells into three broad groups of our granulocytes, our mononuclear phagocyte system, and the lymphocyte population. So for every cell, I want you to ask yourself and keep these in the back of your mind, what does it look like? What does it do? And is it primarily innate, adaptive, or does it help connect the two? Is it that bridge between the innate and adaptive immunity? So, neutrophils should hopefully be familiar from hematology. They really make up the largest percentage of circulating leukocytes and are major phagocytic cells. Think of neutrophils as those rapid response cells, especially when we're dealing with any type of acute inflammation and many bacterial infections. Then we have our eosinophils on the bottom here, which are less numerous. Their major associations are with parasitic and allergic responses. Again, so I don't want you to memorize the percentages alone. I want you to understand and connect that clinical meaning. So, neutrophil, phagocytosis, rapid response, bacterial infections, eosinophil, parasitic, allergies. And again, if you're not familiar with the morphology or if you need a refresher, this one on top is our two neutrophils, and this cell on the bottom is an eosinophil. Then we have our basophils and mast cells, and they are related functionally, especially in allergic and inflammatory responses, but they're not the same cell. Our basophils circulate in blood and make up less than 1% of circulating leukocytes. And you definitely will see that percentage is significant when you are in hematology and you're trying to find a basophil. It's very difficult. And then our mast cells primarily reside in the tissues, particularly places like our skin and connective tissues. So both can release inflammatory mediators such as histamine. So again, if you're trying to distinguish the two, a helpful starting point is to think B, basophil blood, and then mast cell tissue. All right, then our monocytes. Again, monocytes circulate within the blood. But when our monocytes then migrate into tissues and differentiate, that's when they become macrophages. So it's a really important blood versus tissue relationship. And I want you to notice like the morphology. These are large cells with that characteristic horseshoe or kidney shape uh, nucleus, but their importance really goes well beyond the morphology. So macrophages are a perfect example of why innate and adaptive immunity cannot be completely separated. On the innate side, macrophages phagocytize microorganisms. They kill microbes, they release mediators, and they participate in those inflammatory responses. But they can also process antigen and present it to T cells. So macrophage can essentially go from I found something and ate it to now I'm going to show the adaptive immune system what I found. That's one of our bridges between the innate and adaptive immunity. So if macrophages are an important bridge, dendritic cells are one of our most important professional messengers. They capture materials in tissues and are extremely effective at presenting antigens to T cells. The key term here is antigen presenting cell, or APC. When you hear APC, I want you to think this cell is taking information about an antigen and is communicating it to the lymphocyte. So, our dendritic cells are especially important for initiating adaptive immune responses. So now we're going to move into our lymphocytes. So there are three major populations shown here, and those are our B cells, T cells, and natural killer cells. So morphologically, the B cells and T cells can look very similar on a routine peripheral blood smear. So generally, you can't look at a normal lymphocyte and confidently say that's a B cell or that's a T cell. Instead, we distinguish lymphocyte populations by using things like surface markers, which brings us back to that cluster of differentiation or CD terminology from our key terms. B cells are primarily associated with humoral immunity and the antibody production. Again, B cells mature in the bone marrow, and once an appropriate B cells encounters an antigen and becomes activated, it can then differentiate into a plasma cell. And then that plasma cell is essentially an antibody producing factory. So remember that progression: B cells, antigen recognition and activation, plasma cell, antibody secretion. It's having all the antibody babies essentially. And then notice those CD markers. CD markers are the little molecules that are expressed on the cell surfaces, right? That help us identify and characterize different cell population. And this becomes so important in areas like flow cytometry. And we're really going to start to get into the weeds of CD markers as we continue throughout the course. Then we have our T cells, and again, our T cells mature in the thymus, and they mature in, mature T cells then express CD3. Then we have different functional subsets of our T cells. So we have CD4 positive helper T cells, and they help coordinate immune responses through something called cytokines. And we'll talk about cytokines in a bit. Regulatory T cells can help suppress or control immune responses. And then CD8 positive cytotoxic T cells can directly kill virus infected cells and tumor cells. So here's the shorthand I'd like you to start building. CD3 equals T cell. CD4 is a helper. CD8 cytotoxic. And then we'll add nuances later. So the natural killer cells are so fascinating because they're lymphocytes, but functionally they're considered a part of the innate immune system. They don't require the same antigen specific activation process that our B and T cells do. Our natural killer cells are particularly important for killing virus infected cells and tumor cells. They also express characteristic markers including CD16 and CD56. So here's another place where morphology alone doesn't tell us the whole story. Immunophenotype matters. All right. So before you go, I just want you to think back and challenge yourself. See if you can explain these terms without looking back. So, for example, what is the major difference between innate and adaptive immunity? Which cells primarily phagocytize? Which cell is effective at initiating adaptive immunity through antigen presentation? Where do B cells mature? Where do T cells mature? What's the relationship between a B cell and a plasma cell? And why are natural killer cells unusual among lymphocytes? So again, today wasn't about, or this lecture wasn't about memorizing every single immune pathway, but we did build the framework. We have an innate system which recognizes that danger quickly, right? Then we have the adaptive system, which is capable of antigen specific responses and memory. And then we can have cells that then we have cells that kill, cells that engulf, cells that communicate, and cells that produce antibodies. And most importantly, these are not independent pieces, they're constantly communicating. So, as we continue, we're really going to start filling in the mechanisms that really allow that communication to happen. As always, if you have any questions, comments, concerns, please reach out to me and I'm more than happy to help clarify. Otherwise, take a break or go ahead and head over to part 2.